Thank you, Dr. Arriaga and the organizers. I actually made my title up here a little bit shorter and hopefully a little more palatable for all of us. So I wanted to, and I had to give these relationships to disclose grant support from Novartis and consulting fees from Chugai, Foresight, and Oncotherion. I wanted to give a quick introduction to the role of estrogen in breast cancer. Estrogen has long been known to be the key hormone driving normal mammary development. So if you look at here on this slide, we're just looking at a mouse mammary gland, and then if you take away the estrogen receptor in the bottom, you actually really constrict down the mammary development. And so it's no surprise then that given that essential role just in normal mammary gland development, that estrogen receptor and estrogen is a key for 70 to 80% of breast cancers. So in terms of recent work, we and others found mutations in the estrogen receptor in patients who had advanced cancer that had spread outside of the breast, so-called metastatic breast cancer, and in particular patients who had prior exposure to a class of drugs called aromatase inhibitors, drugs that lower circulating levels of estrogen. So what do these mutations do? These mutations make the receptor no longer need estrogen. The breast cancers seem to become estrogen independent, and that's shown here in this preclinical work where cells can grow despite the absence of estrogen being supplied to them. So that led us to kind of two major questions as we were thinking about this for the clinic. How often do we see these constitutively active estrogen receptor mutations in patients? And then what are the outcomes? How do patients fare who have these mutations present? Do they do better than patients who have no mutation? Do they do worse than patients who have these mutations present? To answer that question, we went to a already reported out clinical trial, the BOLERO-2 clinical trial, because it enrolled a large population of women who had the key features. They had estrogen receptor expressing breast cancers that had spread outside of the breast. They were metastatic breast cancers, and all of those patients had previously been treated with the drug, the aromatase inhibitor. And we were able to analyze samples from 541 of the patients on the trials, those who consented to this type of analysis. And what we did was we actually looked at the plasma, not the tumor itself, but the plasma that was taken at the time that they entered into the study. And we looked for the two most common mutations in the estrogen receptor that we'd previously found in 538 and 537. And so one of the key findings of the study was shown here. 29% of the patients had one of these two mutations. 15% had the 538 mutation, 8% had the 537 mutation, and 5.5% actually had both mutations. So this is a common alteration in patients with metastatic breast cancer. The second thing that we did was, well, what we typically do in genomically sort of driven cancer medicine is we take the tumor and look for mutations in the tumor. And to get the tumor, we kind of go back to maybe their initial breast cancer in the breast, or we go to a biopsy from 2 or 3 years ago, and we ask, are the mutations present there? And that's kind of a standard that's done for a lot of clinical trials and was, in fact, collected for this trial. So we used that part of the analysis to compare our test, which was a plasma-based test, with the biopsy of the tumor-based test. And what we found was the mutations in the biopsy of the tumor were rare. Only 1% of the tumor had it, but 30% of the plasma at the time they entered the study had it. And we can talk later about why the reasons why that sort of discrepancy might be present. But it did tell us that plasma was finding a lot more mutations for some reason. Moving on to how did the patients fare? So we know the mutations are common. How did the patients who had these mutations fare compared to those who didn't? And shown here are the survival curves looking at how long just women who had these alterations or not lived. And the median overall survival was significantly lower for those who had either mutation present. And that's just comparing the yellow versus black on these curves. And if you look at each individual mutation, the 538, the 537, or both mutation, each of those groups had a shorter median survival than those who did not have any mutation present. And all these were statistically significant. So immediately that told us that the patients who had these mutations had a worse prognosis than those who did not. Finally, in this very sort of hypothesis generating an early kind of look at what else we might learn by looking at these mutations, we looked at what happened on the trial itself. So the trial itself was looking at giving those patients who had metastatic breast cancer and previously treated with an aromatase inhibitor, we asked what happened if you gave them a different type of aromatase inhibitor, or that aromatase inhibitor plus the mTOR inhibitor everolimus. And that was the trial showed that the addition of everolimus significantly increased how long patients were on therapy, median progression-free survival, and led to the approval of everolimus by the FDA. So we looked at how did patients who had mutation do on these two specific regimens. And just shown here is one example of that, where we looked at the addition of the everolimus and asked how did the wild-type group do and how did the mutant group do. And what you can see is most patients benefited by adding the everolimus. That's the black to red on the right side. You see a large increase in the median progression-free survival, meaning how long they were on the therapy, a 4.5-month increase by adding everolimus. That was also true for the group who had the D538 mutation. But unexpectedly, the group that had the Y537 mutation did not show this benefit. This was a small group of patients in the Y537 group. It's not something that we would now say don't go out and give today based on this result. But it does tell us the biology, and what we need to do going forward in the clinic is look at these mutations, not just as a whole, but individually, and ask how do they impact therapies that we're developing going forward. So I think a sort of hypothesis-generating result as we look at how these mutations might be impacting therapy in the future. So with that, I just want to reiterate a couple of the conclusions. First, that we can detect mutations in the estrogen receptor pretty readily using plasma analysis. These were actually plasma samples that were collected not with the intent of looking specifically for estrogen receptor mutations, but they were collected as standard plasma was, and we were able to use them for this. Second, mutation was quite frequent in this population. 30% at least have mutations in the estrogen receptor. Third, by using plasma, we actually think we're doing better at detecting mutation than if we go to old archival tumor materials. Fourth, patients who had mutation had a shorter median survival than those who did not. And fifth, the patients with different mutations might end up having different responses to therapies in the future, and I think that's an important area for future research. So with that, I will welcome questions.
Thank you. Thank you, Sarat. May I ask the first question, if I may? So if I understand correctly, these patients had the mutation detected at the time they had metastatic disease, but there was a discordance with the biopsy of the primary tumor that may have been done years before. Can you speculate as to the reasons for that discordance?
Yeah, so we think that there might be three different reasons why the plasma at the time they entered the study is picking up mutations more frequently. One, the archival tumor is often the primary breast cancer, and we've already observed that with metastases, cancer that's spread outside of the breast, there's more frequent identification of mutation. By virtue of the study, we're only looking at the time that they had metastatic disease when we're looking in plasma. Second, primary tumors or archival tumors often were collected prior to the exposure to that drug, the aromatase inhibitor, the ones at lower levels of estrogen. So there's a selection over time for these active mutants when you give them the drug, the aromatase inhibitor. And so what we're seeing is after they've been exposed to the drug, there's a higher incidence of the mutation. And third, when we do plasma, we're looking at some sort of summation of different metastatic or spread breast cancer. We're looking not just at one particular lesion that we biopsied, but perhaps many different lesions. So if a woman has a liver metastasis and a bone metastasis, we often only sample by biopsy one of those, but by plasma we think we might be capturing the summation of both of those. So I think for all these reasons, plasma may have certain advantages, especially for this alteration.